This video is going to model the process of mitosis with a cell that is 2n equals 4. That means there are 4 chromosomes and they're organized in homologous pairs. So this is a homologous pair. They are the same length, they have the same number of genes, they have the same type of genes. So for example, this might be the same gene, but there are different forms of it, so they're different alleles represented by different colors. Here's another homologous pair, again, same length, so same number of genes, same type of genes, but the type of information or the allele might be different on the 2 chromosomes. So that means there are 4 chromosomes and they're organized in pairs. That's what that 2n means, is that they're homologous pairs. All right, so before mitosis or meiosis is the process of DNA replication. And the DNA is going to replicate for each chromosome identically. And this white little piece of paper here represents the centromere, and that connects the sister chromatids together. So these are 2 identical sister chromatids attached at the centromere, and we'll do that for all of our chromosomes. And this is where you see this characteristic X pattern that you see in a lot of diagrams. Keep in mind, this X pattern is only true after DNA replication. So mostly our cells don't look like this. Mostly they just are those single strands. And in fact, the chromosomes aren't even condensed for most of our cells' lives. All right, so after the DNA is replicated, then we can start the process of division if everything proceeds through the cell cycle. So if we get through the cell cycle and we get to mitosis, right, so the first step of mitosis is the prophase, where we start having the breakdown of the nuclear envelope. The next step is going to be metaphase, where everything, all of the chromosomes line up on the metaphase plate. Let's see if I have enough room here on the video to show all of these. Maybe not quite. I'll maybe move these out of the way here, give a little bit more room. 1, 2, 3, and 4. All right, so after metaphase, after they're all lined up, then we get anaphase, which is the separation of these chromosomes. So those sister chromatids start pulling apart. Okay. And the next stage after anaphase is telophase and cytokinesis, which is where we have the full division and the reestablishing of the nuclear envelope, and the cells are completely separated. The cells are completely divided. So at the end of mitosis, we end up with 2 cells. And if you notice, these 2 cells are identical to one another. They both have 4 chromosomes, and those chromosomes are organized in homologous pairs. And they are also identical to the cell that we started with. So this is useful for growth and repair, where you need more of the exact same cells with the same type of, with all the same genetic information. This is also good for asexual reproduction. Like, for example, single-celled organisms would go through this division in order to make then 2 individuals. All right, so that's mitosis. In this video, we're going to model the process of meiosis in a cell that is 2n equals 4, which means it has 4 chromosomes, and the 2n signifies that they are organized in pairs. So you can see we have 1 homologous pair here and 1 homologous pair here. So there are 4 total chromosomes, and they are organized in pairs. All right, so before we undergo meiosis or mitosis, the first thing that happens is DNA replication. So we get DNA replication is the identical copying of each of these chromosomes. At the end of this process, we still have 2n equals 4. We still have 4 chromosomes, 1, 2, 3, 4, and they're still in homologous pairs. This is a homologous pair and this is a homologous pair. The only difference is now they're in their replicated form. So we have 2 chromatids for each of our chromosomes. All right, in meiosis, there are 2 division steps. So the first division step of meiosis is called Meiosis One. And in this step, we get our homologous chromosomes separating. So let's go through all of these steps. So the first step of Meiosis One is crossing over. So we have our homologous chromosomes get near each other, like this and like this. And you'll notice this paper is representing the centromere. So the sister chromatids are attached by the centromere, and they line up, homologous chromosomes line up with their centromeres near each other. All right, so crossing over is an exchange of genetic information between non-sister chromatids. So for example, right here, we maybe have this crossing over here. And if it crossed over here, we would have 2 beads on this one, swap with 2 beads on this one. And we're just going to represent in this model the crossing over happening on 2 of these non-sister chromatids. All right, over here, so we've got kind of one X shape here, one X shape here, and then we'll have crossing over. Now, crossing over might happen in more than one location, but we're just in this model going to show it happening in one location. So maybe it happens 3 beads in over here. So here's crossing over, and we split this up. So we take these beads off here, these beads off here. All right, so this is now showing after that crossing over step. All right, so we had an exchange of genetic information between non-sister chromatids. Right, the next thing that happens is Metaphase One, where we have our homologous chromosomes lining up on the metaphase plate. So we've got one homologous chromosome there, one pair of homologous chromosomes here. And then in the next step, after they're all lined up, then we get Anaphase. And this is when the homologous chromosomes are separating, Anaphase One. So these start heading this way, these start heading this way. All right. Next up, got telophase and cytokinesis, and this is where the cell actually divides. So now we've got a cell over here and a cell over here. So after Meiosis One, we now have 2 cells, this cell and this cell. In each of these, n is equal to 2. We have 1 chromosome here, 1 chromosome here, but they are not a homologous pair. And so that's why it's not 2n anymore, it's just n, because these are not a homologous pair. So 2 chromosomes over here in this cell, not in a homologous pair, means this cell is n equals 2. And over here, we've got 2 chromosomes, and they are not a homologous pair, so this cell is also n equals 2. All right, so that is the end of Meiosis One, which means now we're going to have Meiosis Two. All right, first step of Meiosis Two is prophase, where the nuclear envelope starts breaking down, and some other details we're not going to go into. Metaphase Two, here we have the lining up in each of our cells. Remember, this is a cell and this is a cell. So we have the lining up of all of the chromosomes on the central metaphase plate. Okay. Then in Anaphase 2, the sister chromatids separate. So we have these going this way, and these start going this way. Over in this cell, these start going this way, these start going this way. And then that separation is finalized in telophase and the next cytokinesis. And so now, both of those 2 cells, this cell divided into 2 and this cell divided into 2. So at the end of this process, at the end of Meiosis 2, we have 4 gametes. We have a gamete here, gamete here, gamete here, and a gamete here. And if you'll notice, all of these gametes are unique, and they also all have 2 chromosomes that are not in pairs. So here we have 2 chromosomes not in a pair. And so each of these is a haploid gamete because it has half the information of the original parent cell. And that is meiosis.